Hey everybody, welcome to Town Hall. Thanks for joining us again. We're just going to do a little small talk while people join up. The good news is Joe has his coffee, so we are all in good hands in that sense. And I just want to note that the sun is out because it feels like it's been a little while since we saw it. So my weekend, I don't know if it's also going to be, good and cold. I'm going to be doing some household improvements, like shades and
Hmm.
things like that. We're still like making an apartment livable. Anybody else have plans for the weekend as we wait for people to join us?
I'm
Getting it.
supposed to go fat biking.
Ah,
Oh,
Yeah,
oh, good for you.
that's very fun.
Where does one do
Uh,
that here?
so we're probably gonna go up to Silver Lake Park in Wisconsin. They've got, bike trails, or there's a place, Paul Douglas, out in Hoffman Estates, that's all mountain bike, single track bike trails. So,
Wow. And
and
so, what's your frostbite plan?
I just bought a new box of hand warmers and foot warmers, and believe it or not, you get hot pretty quickly.
But
So,
I'm thinking like nose.
oh yeah, no, we
Okay.
have got like face
You got
paint.
face things. All right.
Yeah.
Anybody else doing anything dramatic and limb threatening for the weekend?
hopefully not limb threatening. No, a pre-cut tree, so no chainsaws involved.
All right. All right. Elise, anything?
The my PI is here. Friendly response is working on our paper, which could be limb threatening, depending on how.
All right.
But
Well,
no, I'll be at home, hunkered down.
all right. Well, so I want to welcome our panel. We've got Joe DiMario, who's our Executive Vice President for Research, Beth Stutzmann, Professor, and she's Discipline Chair in Neuroscience, and Elise Weber, who is a PhD student with us today. I'm just going to do a little bit of sort of an update, president's update, and then we'll move to Joe and talk a little bit about science. So this week, the board had its finished up its subcommittee meetings, and we had the actual board meeting. It was virtual, but they approved our budget outline as a part of the 5-year budget plan. This is essentially a top-down estimates of how much revenue we think we're going to have, estimates of what kind of expenses we think we'll have. In January, probably late January, Gavin Ferry will come on to a town hall and talk about the process of the bottom-up budget, which is actually how we create a budget. And then later on in the year, when we know more about what our enrollment will look like, we finalize the budget. The thing goes to the board for approval. So that's just a little bit of process. So we had a group of promotions that were approved by the Board of Trustees. That's always a fun and wonderful thing. Our team gave a bit of a presentation on early start and the Grad Plus loan situation and things like that to the board to help them understand the uncertainty that our folks will be facing, the students and our programs, as we look to what's going to happen to federal funding of student financial aid. So it's a big, complicated thing, but I really appreciate our team and all the work they've done, all the faculty and staff who've been working on early start. It's a huge lift, but also I think really important for our incoming students. And then the backside of that is that applications are up, so which is great. So I have a few numbers. Chicago Medical School is up 38%. I happened to get some mail yesterday from the Association of American Medical Colleges. Average across all medical schools is only 6%. So CMS's 38% is a big jump over what's happening nationally. The PA program is up 61%, CRNA is up 75%, and PharmD is up 42% on applications. That doesn't always turn into students, but I think it's a great sign for what our folks have been doing, what our people in strategic enrollment management and marketing have been doing, just to help prospective students and their families understand the value and great experience that students have at Rosalind Franklin University. Also this week, Tolmar Pharmaceuticals attempted to cut a ribbon and eventually ripped a ribbon to enter the Innovation and Research Park. They did actually. It was a ribbon cutting ceremony. It was a lot of fun. And I got to learn a little bit about what they do. So they tend to take drugs that are on the market and then reformulate them to have a uh, longer time between administrations. So, for example, they use loperamide, which is uh, a drug that you might know as Lupron, which is a hormone blocker, and it a part of what they do is they formulated a version that can be used for precocious puberty twice a year. And so because of the essentially the compounds that they stick onto the actual medication, it takes a while for the body to liberate it, and that means you can go much longer between doses, which makes it a lot easier for compliance and often toleration. So it was really interesting to meet their folks. This is one of the important things for a university to do in a community. So Rosalind Franklin University has about $285 million of economic activity in total. 265 million of that is in Lake County. Lake County is a core biomedical center for the Midwest and the country, and it's important that we play a part in that. We are not only about providing educational and care and other kinds of outreach, but also economic outreach. That is a part of what universities have traditionally done, and it's important that we're a part of that. We had our health equity breakfast because, of course, economic activity doesn't get everywhere it needs to be. Beth was one of the panelists at our health equity breakfast. I focused on neurodegenerative disorders. It was there. Really, our foundation funders were there. It was great to get a chance to see them and hear about some of our work. And then I got to have some fun this week as well. I got a little bit of a tour of the new virtual hospital and clinic. They're still they're the I let's see here. The builders were there, and I got to meet the people who put in the sprinklers and things like that. So it's getting closer. But then I also got a chance to go to the turkey dinner last Friday night over at the pavilion with about 40 students or so. Got to drop in on the CMS holiday party, the marketing party, and I had a lot of fun in cafeteria time where I just walked down to see students. So anyway, I've had a fun week. It's been great to meet even more people, which is my time to warn Joe that I'm going to turn it over to you here in a second. But anyway, I just appreciate how friendly and welcoming everybody has been. I am trying to learn as much as I can, as quickly as I can, which has been a lot of fun also. But one of the things that I enjoy most is getting to hear about the work our faculty and students do. That's one of the reasons for doing a town hall is for I can share some of that experience with you all. So Joe, this is really a time, Joe's going to talk a little bit about research in general. We're going to move over to Beth and Elise for their work in more specifics. So Joe, the screen is yours.
Thank you. Thank you. So let me bring up my PowerPoint here. It'll be short.
As Joe's doing that, I want to remind people this is more fun with questions. So whether it's about what I talked about or Beth, Elise, or Joe, feel free to put your questions in the Q&A.
Absolutely. So the first thing that I really wanted to do today was give a shout out to everyone that contributes to research at RFU. And I'm just going to flash this up. I'm certainly not going to go through it all. These are research centers throughout the university, spread across all colleges, schools throughout the university. So just a shout out to all of the leaders of these centers shown on this slide. And as I said, the research is spread throughout all the colleges and schools. There's tremendous high quality research everywhere. And I just wanted to take this opportunity to say thank you to everyone that contributes to it. In addition to those centers, there are quite a few of support offices and committees. And so just from EHS, IACUC, the IRB, OSR, all the vegetable, or excuse me, all the alphabet soup that you can think of. Office of Research Integrity, Institutional Biosafety, research data, all of those activities are going on in the university. And again, thank you for everyone that contributes. And then lastly, we have a whole host of very good, high quality support labs, core facilities managed by the people that you see on here. Again, too many to shout out individually, but just my sincere thank you to everyone that contributes, as well as everyone that contributes that I cannot put on PowerPoints because it's too many. All the people in the research centers, the staff that contribute to our research administration.
One of the things I wanted to just, as you come to that, one thing that people often don't know, because there've been a lot of conversations about indirect cost return and things like that from the NIH, and people don't often realize that there are more than 250 regulations that we have to be compliant with and demonstrate compliance with to get federal funding, which is a part of the reason that, it is so much work to run a research operation.
Yeah, just OSR deals
Hmm.
with a tremendous amount of regulations, and those regulations are changing pretty quickly recently.
Hmm.
And so Dora Espinoza and the staff within that office do a wonderful job of keeping updated, keeping me and the rest of the university updated on all these changing regs. What I also wanted to do was just flash up a few numbers here. This graph shows the amount of extramural funding that comes into the university, broken down by total research dollars in the red columns, and in that sort of tan color, federal dollars that come into the university. And you can look at the net amount there of hovering around 10 million or so last fiscal year. But what I'm most interested in always seeing is the upward trend in securing extramural dollars coming into the university. So we're on a good trajectory, and of course our one of our jobs is to try to keep that trajectory going as best as we possibly can. Just as an additional information, our offices throughout the university track a number of key performance indicators, and all of those consistently look good in terms of research dollars and expenditures and finances. Oops. We continue to invest in new researchers coming into the university through salary and startup packages, and we've had a number of recent hires. Dr. Arneson Wisseg actually isn't here quite yet, but she signed a letter of intent to come into the university, and we're looking forward to her coming in probably in the first quarter of next year. She works on inflammatory signaling and cancer cachexia. And I'm particularly interested in talking to Paige because she works on the effects of cancer on skeletal muscle metabolism, an area that I've always been interested in. So we met for a brief time, but it was a wonderful conversation just about science. Something always wonderful to do. We also do have additional recruitments going on in the Center for Genetic Diseases and Cancer Cell Biology. So we continue to invest in new, exciting research and faculty coming in. And then I know there's always interest in what's happening with the IRP and Helix 51. Shown in the upper right are current tenants in Helix 51. UP Oncolytics, for instance, is a company that works on viral administration of vectors to combat various types of cancers. They recently have secured other funding, so their prospects look very good. Monopar Therapeutics is a relatively recent addition to Helix 51. In fact, Helix 51 is doing so well that the university continues to look for additional space to house small startup companies that are interested in coming into the Helix 51. So that Helix 51 is more of an incubator type setting for small proof of concept type companies that want to come in and establish their research. A new addition to that is a company called Deep Brain, where they're exploring novel drug delivery techniques to fight various forms of cancer. The IRP, Dr. Sousa mentioned Tolmar as a resident in the IRP. The ribbon cutting was for their recent occupancy, and they're also planning to expand within the IRP in the near future. So we're excited to have them as a tenant. You can see some other tenants and the types of activities that go on in the IRP range from biotech, biopharma to medical devices. And we've recently come to an agreement with an organization called the Chicago Clinical Research Institute, which performs phase one clinical trials. So we've expanded the types of organizations, companies that can come into the IRP, and we're seeing now increased occupancy so that by the time CCRI, as well as perhaps another patient-based organization comes in, the IRP will be well over 50% occupied by tenants, and many of them have long-term leases. So we're excited to see the progress of both Helix 51 and the IRP. With those good news, there are also some challenges facing research, and many of you have probably heard of this. Dr. Sousa alluded to indirect costs, and so the U.S. Congress right now is considering some alternate models to fund finance and administrative costs, otherwise known as indirect costs. And again, these are costs. These are costs that universities incur for offices that I just showed, like OSR, EHS, all of the activities that are required to support research, and many of which are federally mandated. But nonetheless, they are not covered by direct costs, and so we try to recover those costs through indirect rates. We'll see what Congress and the federal branch what agreements they come to. That has not yet been formally deliberated.
Yeah, it's actually
Congress,
and these
yeah,
are the indirect costs are things that we negotiate with the federal government generally. They are it's actually reimbursement. So the whole process when you set up your IDC rate is a reimbursement question. How much do you actually spend? And
yeah,
it includes space and a whole range of other things that foundations don't often include. So
right,
it's not only big, complicated, and consequential, but it's not like it's free money. It's money we've already spent. They're paying us back.
correct, correct. So right now our rate is 57.9% for federal grants, and OSR approximately every month will request those funds from the federal agencies to be sent back to Rosalind Franklin. So as you said, yes, we've already incurred those expenses. And when things occur like government shutdown, other federal irregularities, I know the finance department is always anxious to be able to recoup those funds from the federal agencies. The other thing that's going on at the federal level is a discussion of the funding of the federal agencies themselves, so NIH, NSF, DoD, et cetera. Both the House and Senate have proposed increases to the NIH budget. The President's office and administrators are proposing a cut to federal agencies, and so we are awaiting the resolution of those discussions as to how it's going to fall out. You may have heard of 40% or so proposed cuts to the NIH budget, but again, those negotiations are going to have to work out between what Congress wants to see and what the administrative branch wants to see. Have also been reprioritization of federal research aims to highlight aspects of research like nutrition, chronic diseases, effects of environmental factors on disease, et cetera. So I know that our researchers are considering ways in which they can readjust their research portfolios, their specific lab research aims to better align with these new federal research priorities. And then the last item on this list is the near-term effects of the government shutdown. Because of the government shutdown, there were no federal agency meetings, so study sections didn't meet. And so there is a tremendous backlog now of study sections, and the effective upshot of that is that triage rates will increase from approximately 50% to 65, perhaps even 70%. And the long-term negative effect of that is for those grants that are triaged, investigators get less of an informed opinion as to how to improve their grants for resubmission. And so overall, it becomes more difficult to secure funding based on the government shutdown. And so in response to this, some of the goals in research for the remainder of the fiscal year is to, first of all, improve the stability and growth of research. And part of that is going to be by building a comprehensive 5-year budget plan that incorporates operations, capital expenditures, as well as new faculty recruitments. And those will be aligned with university strengths, university needs, as well as federal priorities. We're always looking to further develop and implement plans for philanthropy. Dr. Sousa alluded to the breakfast and health equity this week, and so there's already been a little nibble of activity based on that. So we'll try to take advantage of that. And then we'll continue to try to foster bringing in biomedical companies and collaborative partners into Helix 51 and the IRP. And then continue to work on the federal challenges in terms of indirect costs, federal reprioritization of research, and working with all the offices, investigators throughout the university to try to keep our research program going. So I
Great.
know since I can't
Yeah.
see
So
hands.
we have a few questions. So let me get through some of those, and then
Yeah.
did you have more before we go to Beth?
Nope, that's it. That's it for
Okay,
me.
so Carl Correll asks, when writing a letter to our congressional representatives, what points should we emphasize to encourage them to reverse policy that denies federal loans to students in nursing, PA, PT, etc.? I realize that when I write this letter, it must be as a constituent, not an RFU employee. Points that I think of are shortages in these professions, their essential roles in healthcare, the fact that they are board certified, that these professions help make healthcare more affordable. Any suggestions? And I mean, I think Carl has actually done a great job of outlining some of those points. Uh, this isn't. I think of these programs as in 2 or 3 different ways. One is it provides an opportunity to people, and if you want to support first generation students who come from a disadvantaged background, then having a way for them to be able to afford to borrow the money is really important. So whether you're from a rural community that hasn't had great high schools or an urban community that faced similar situations, you're in a disadvantaged status, and often the federal programs help those people get that opportunity. And the second is that we absolutely need these professionals to help provide care. And I think it's kind of shocking to me that nursing is not considered a profession in the federal eyes. It's a graduate program. So I think that you can simply point out that they're expected to behave as professionals, and the federal government should treat them as such. So in any case, we're a nonpartisan organization. Contact your folks as you will, but there's no doubt that if you're interested in providing opportunity and providing health care, particularly in communities that need service, the current federal policy is not supporting that. David Muller asks, if IDC decreases, will you get the money by direct route, such as charging rent and tuition? And so that is one of the arguments or questions about how IDC rates might change, that some of it might switch over to direct cost. So you would be able, right now in a lot of foundation grants, you can actually provide a charge for accounting and put that on the grant. And so we can't do that with the NIH, but inconceivably you could. The same thing for space. Right now, one of the reasons that, places like RFU and many places don't have charges for space to faculty is that they don't have charges to space for the feds. So all those things have downstream consequences for us conceivably. But yeah, direct costs are certainly one of the ways that the difference could be made up. And then Anonymous asks, is there an estimated timeline for hitting 100% occupancy at the IRP? Joe, can you help me with that one?
Yeah, I would say, obviously difficult to predict with certainty. But I would say over the past year, there's been a marked increase in the interest and occupancy rate within the IRP. So while I hesitate to put a specific date to it, I would anticipate that with organizations that we're currently in discussion with, really the significant majority of the IRP, I would think would be full within the next year or so. Um, Barring turnover companies, leaving, et cetera. We've already had companies that have been in the IRP leave the IRP. So now we're also reaching the point of turnover. But
Which has to happen.
yeah, right. They're businesses. Sometimes businesses succeed. Sometimes they
Yep.
don't. Sometimes they expand. Sometimes they move for their
Yep.
own priorities. But nonetheless, in my estimation, the IRP now has really turned a significant corner and is expanding quite well.
So we have a couple more. Jeff Lang asks what percentage of the IRP is currently occupied.
Yeah, well over 50%. And I would
Okay.
anticipate within a year's time, probably on the order of 80,
Okay.
85%.
Charlotte Beyer asks, one of the challenges faculty have raised are the challenges in federal changes and requirements of making research open access and who pays those fees. Is there a university plan to support researchers if they need to pay the APCs? And I don't know what RFU has been doing, or what the library's piece of that has been, but Joe, do you know an answer to that?
I No, I don't.
Okay.
No, sorry.
It's
I'd
it's a
have to look
problem
at it.
nationally, right? And often it has to come off of grant dollars or other dollars, but it is certainly a one of the things. While having things open access certainly makes a lot of sense. It is an opportunity for sometimes for journals to crank up the price, and it's an additional expense for our researchers. Mark Glucksman asks, is there a webinar? There is a webinar at RFU noon on Tuesday, the 16th, on advocacy and an update on Washington's going on given by our own Nick Caliaris, the VP of Government Relations. That's a key little piece. And then David Harrison asks, why are non-federal dollars down since 2021?
I'm going back to that slide. Non-federal dollars in the graph that I showed continue to increase from 2022 up through 2025. There
Okay.
was a time before this slide that some of you may recall that the numbers had gone down, and that was sort of the hangover from COVID. The graph that I showed shows increasing federal dollars for both total and federal. If you're referring to the difference between the two, I think it's simply maybe an emphasis that we're trying to focus on NIH dollars, federal dollars that are coming in. That, though, may need to shift over as federal dollars may become more limiting. So our faculty are very much aware of the opportunities that currently exist for private foundation funding, but they are also going to be slammed by requests from researchers throughout the country. So in both respects, both federal and private foundation funding, competitiveness is going to increase.
Well, especially that triage rate, right? Which means that fewer people will get
Right.
pink sheets, and feedback from grants means that, it'll be harder for people to start up new projects and for new faculty to get their first grant, which means that foundations will perhaps be even more important for pilot data and getting started and things like that. And Michael Rosen points out, Michael Rosen, who's is on point for helping us find companies and get them into Helix 51 on the IRP, points out that the clinical immunology lab will be moving in to the first floor of the IRP in the first quarter of 2026.
Correct.
Questions always make this more interesting. Feel free to keep putting them in, but I think this is our time to move to Beth. So Beth, this is a chance. Really, I wanted to have a faculty and student talking a little bit about their work. So Beth, the screen is yours. You are muted, and this is your presenter version.
0 for 2. Let me try something else. Let's see. Usually sharing is not my problem, so I don't know why this is being difficult. Let me try this one. This is the entire screen, content only. Let's see.
Yeah, that button right there.
There.
Oh, but it's giving us presenter again.
Oh, that's so annoying. I don't know why it's doing this.
Yeah. If we can
how
live
about
with
we're
this.
just gonna wing it?
Yeah.
Can we just do this?
Yeah, we can live with this.
All right, I was gonna say, I'd rather just move forward.
Yep.
Yeah, but like, thank you very much for inviting me to chat about some of the work going on in my lab and in the center as well. And hopefully I want to highlight some of the really exciting work the students have been doing. The crop of students I have are amazing, Elise being one of them. But I got to get through a lot here. I did peek through the list of attendees and I saw that there was a wide range of backgrounds from, clear like non-scientifics, essential support staff to, extremely smart physicians and PhDs. So I'm going to try and land somewhere in the middle, but I do love questions. I love to talk about this stuff, as you will quickly see. So please barrage me with questions. My intent was to give sort of a broad overview, but if anyone had interest in a particular area, definitely circle back to me in that. But really what our lab has been doing for years is trying to understand the cellular and circuit level mechanisms that are going wrong in neurodegenerative diseases like Alzheimer's. And many of you have probably heard about some of the key features of Alzheimer's disease, like amyloid plaques and things like that. We don't focus on those per se. Actually, Bob Marr is very much more of an expert in that area. I'm more mechanisms, cellular physiology, synaptic memory encoding stuff. But really with Alzheimer's, we know extremely little about what's causing it. I'm kind of going to skip over what it is since most people probably know. But age is one of the major risk factors, but that's about all we got. We do not know what causes Alzheimer's. So we're trying to approach this from a different way and look at two of the major risk factors for Alzheimer's and then kind of overlap them to see, well, what shared features do they have that could inform on the mechanisms of Alzheimer's disease? And so for this prior brain injury is one of the big epigenetic risk factors, and then Down syndrome is the #1 genetic cause of Alzheimer's. And that's a very interesting story unto itself that the lab is really doing a deep dive into. So we're sort of triangulating these things and trying to pull out common denominators. And we knew at the most basic level that there is an enormous amount of shared differentially expressed gene pathways and microRNAs that overlap. And if you condense those into the major functional consequences, and our lab really is more of a functional physiology lab, they fall into 4 neat categories, all of which we have had some expertise in studying. So intracellular calcium signaling and handling, and this drives so many things in cells. And for those that aren't into second messengers, I'm not referring to the sort of calcium or the calcium phosphate that's in bones. This is free calcium, the ion that's basically serving as like a little signaling factor. There's also the endolysosomal pathways, autophagy pathways, the ways in which cells, and especially neurons, since they're non-dividing, clear debris and unwanted protein products like the ones you find in Alzheimer's. Also, mitochondrial deficits, so powerhouse of the cell, bioenergetics, all of the genes that are controlling the oxidative phosphorylation pathways and bioenergetics. Again, all those genes are differentially expressed in those conditions. And also synaptic signaling, which is the one that's nearest and dearest to my heart. That's where memory encoding is actually happening. That's the feature that people care about probably most in these conditions is the memory and the cognitive deficits. So all sorts of genes and pathways converging on synaptic signaling are also affected. And so we study this in a variety of different ways. I said we like to study things in living cells or living slices. And we combine techniques such as like whole cell patch clamp electrophysiology with a variety of optical imaging techniques such as tooth photon imaging or CMOS microscopy, and then deliver drugs or compounds or stimuli in a variety of ways such as UV photolysis or genetically encoded biosensors. And then we can measure all these things simultaneously. So this is an example of a neuron we've patched onto, filled with a calcium indicator, and we can release calcium from intracellular stores and measure those responses. And we can measure them in individual synapses and spine heads and things like that. It's really, really fun to watch it happen. We used mouse models of these conditions, and then increasingly, neurons derived from these patient populations, particularly for the sporadic and familial Alzheimer's and Down syndrome. Traumatic brain injury, not so much. That's largely a mouse model utility that Elise will be telling us more about. But these are the sorts of things we do in the lab.
Hey Beth.
Mhm.
Oh, okay, yeah. I was gonna ask you essentially about the calcium piece and what you take from it. So it looks like that's where you're going.
Oh, yes, we spend a lot of time on this topic. So,
All right.
right. And this is actually from my postdoc years. This is where we got started, where we can photo uncage activate these intracellular receptors, measure the response. And this is from a normal mouse. And then just hold that in your short-term memory, compare the same stimuli in an Alzheimer's mouse, and you'll see there's just, more red is more calcium. There's a lot more calcium being released within these AD neurons. And this is early. This mouse is probably 3 to 4 weeks old. So like no pathology, it seems fine, its memory is fine, but this calcium signaling thing is happening very early. And this is all well and good, but it's what is this doing is what we're interested in. And we had spent many years looking at the synaptic component of it. And so for those interested in that and the plasticity and the encoding, we have, plenty of papers. I'm happy to push them to you. I wanted to talk about some of the newer stuff that the current lab members are working on. And one of the, I'm very grateful for Sarah and Wasey and Smriti, who's a newer postdoc in the lab, because they have interest in autophagy and lysosome, and then mitochondrial and bioenergetics.
So tell us what autophagy is or autophagy.
Okay, so autophagy. So for those that maybe I'm dating myself, but you think of Pac-Man and it's just kind of running around its cage and it's eating up all of the little, dots.
You're dating all of us. You're dating all of us.
Autophagy is the means by which cells basically clear out unwanted debris, right? So it's really essential for getting rid of, unwanted protein bits, unwanted organelles. It's basically cleaning house. And for some cells that divide, this happens somewhat naturally as cells are dividing, but neurons don't divide. So this autophagy process is really essential for neuronal viability. And these autophagosomes, those bodies that, the little Pac-Man that eat up all the garbage, then are digested by something else called a lysosomes, right? So lysing, splitting. Lysosomes are these really acidic organelles that literally just digest the contents of the autophagosomes. So these two things need to work in concert with each other. And will be a key point in a second is, again, I mentioned that pH of the lysosomes is essential because what's inside needs a very acidic pH in order to work to break down the autophagosomes. So we're going to start with that. And that huge amount of calcium I was just showing you that's being released within these Alzheimer's neurons, we know, and again, this is probably decades of work that we've done, and others as well, is that it's coming from an organelle called the endoplasmic reticulum, the ER. And it's a really big source of intracellular calcium, and it's being released through one of two channels. I don't want to I'll gladly go into an in-depth cell biology lecture, but just for today, it's largely through this channel called the ryanodine receptor. Joe, I'm sure you know about it because it's also big in skeletal muscle. So this excess calcium is being released through this ryanodine receptor. It's hitting all sorts of other targets and disrupting lots of things. But one of the things it's doing is disrupting this lysosome function. There are also, and I'll talk about this next, within the mitochondria, you need calcium for normal bioenergetics, but when there's too much, it just drives the system into basically an apoptotic or an induced cell death state. And there's a whole series of very specific calcium channels on each of these organelles. Also critical on this lysosome is this proton pump. It's pumping in the protons that keep it acidic. And what helps to balance that is extrusion of calcium. So this will be relevant in a minute. So it's all this fine balancing through calcium. And this ER store here is dumping way too much. So Sarah came in, actually was initiated by Alice Gilman Sachs. Because she looks at one of the proton pumps as well. So we were interested in, well, what is this calcium doing to this lysosome autophagosome relationship in AD? But what I want to take one quick step backwards and like my little snarky comment about how we've already eradicated Alzheimer's disease in mouse models, which is kind of a joke. Mice don't get Alzheimer's. We express mutations that will cause the human form of it, and then we fix it, and then we take it to clinical trial and it fails over and over. So we are increasingly using, and this is with expertise from Bob Marr and also Dan Peterson, neurons derived from these patients, and both sporadic Alzheimer's, which is the overwhelmingly common form, and we get this from the Rush University ROSMAP group for anybody that knows about that. So we also have with these data set, and this is with Justin Wally as well, all this transcriptomic, proteomic, lifestyle, brain sample, histopathology data that go along with this patient population. And now we have their neurons, that we're growing in a dish. So this is a piece of a really important, bigger puzzle. And in these Alzheimer's patients, whether it's the genetic or the sporadic form, we're seeing that same increase in that ER calcium source. And Ryanodex is basically the inhibitor of this channel. So we know that it's predominantly through this ER calcium channel in the human neurons, whether it's sporadic or familial. So we're like, okay, now I feel like we've got some basis to move forward with. And we're not looking at some epiphenomenon in a mouse model. It's not seen. Okay, so moving.
So are these like colonies of immortalized cells, or are they organoids?
So they're not organoids in that this is a two-dimensional cell culture. We could grow them into organoids if we wanted to or needed to. Right now, the neuronal culture, we do incorporate an astrocytic media. So there's components of the astrocytes, but we're not growing astrocytes. And you'll see, like in this picture here, we're patching on to individual neurons, filling them. So we want to make sure that what is in there is predominantly neurons. We're not trying to find neurons within a sea of other cells.
Just for people who don't know what a patch is, I mean, it's essentially a cellular-sized, glass pipette with you stick onto the membrane of the cell, and you can isolate what's happening inside what would be that little hole.
Yeah, absolutely. And since neurons are electrically excitable, we can inject current or voltage and measure current and watch them spike and fire. Again, it's very fun. Once you get there, it's a little frustrating in the learning curve. But yes, thank you. Yeah, please. Like if I'm already jumping over or kind of going off on a tangent, please pull me back, Dr. Sousa or somebody, or Elise. Okay. So the basic foundational biology is there between the human and the mouse condition. And again, with this, the first pathway I wanted to show you was this work by Sarah and Sarah Mustele. And what she found, I know there's a lot going on here. So we can measure the pH of these lysosomes, right? There's biosensors that you can express or put in this dish containing these neurons. And you can measure and calibrate what the actual pH is, right? And we know, and so non-AD, that's our control healthy neurons that we're growing. And this is showing the appropriate acidic pH that the lysosome should be. But in our Alzheimer's neurons, it is really alkaline, very, very alkaline. And so this is, we're wondering, well, how do these proteases work if it's not in an acidic environment? And something else that we thought was really interesting is that so here people see caffeine. It's not the caffeine. I mean, it is the caffeine in your coffee, but it's caffeine is used as a trigger or an agonist to release calcium from that ryanodine channel. So here we're using it as a way to release calcium from that ER channel. And we can take a normal acidic lysosome and alter its pH so it looks like an Alzheimer's one, just by releasing too much of that calcium from the intracellular stores. We can also take that really basic pH, the alkaline pH of the Alzheimer's one, and restore it back to the normal healthy version by blocking that channel. So by either activating or blocking the channel, we can convert one to the other. So that was really, again, a very interesting finding. And again, we've gone into some mechanism, but I only have, 2 hours?
Yep.
And so when we look at the protease activity, right, this is the enzymatic activity needed to digest those autophagosomes. We're losing that protease activity in our Alzheimer's lysosomes. and getting it back when we block that channel. So again, this is all being mediated through that calcium pathway. We're like, well, if the proteasomes or if the proteases, those enzymes can't digest the autophagosomes, what's happening to the autophagosomes, right? Those are the things that are actually chewing up and clearing that debris. And so we can measure the autophagosomes, and you can see here, there's more of them. They're accumulating. They're not being digested, and they're beginning to clog up our neurons. And again, by normalizing that calcium, we can restore the protease activity, clear those autophagosomes, and bring them back to a level, again, where they're being digested. And one of the things, particularly for Alzheimer's and also brain injury and Down syndrome, is intracellular accumulation of this pathological phospho-tau species, right? So it's one of the sticky bad proteins in these diseases, phospho-tau. So what is in these autophagosomes, right? Is it not clearing this phospho-tau? And we certainly see more of it when we're looking at co-localization of the phospho-tau within these AD neurons. And again, normalizing this calcium restores this whole pathway, and we reduce the hyperphosphorylated tau. So again, we're just trying to stitch together the pathological pathway of these key proteins, but why are they there? How are they getting there? The other aspect or side of it, and this is Wacy Gallegos and Schmitty, who came in with these interests and expertise. So I appreciate these students bringing in these ideas and tools and techniques, because I wouldn't have gone down these pathways. And Sarah and Wacy and Daniel Steinbrenner and Schmitty in the lab, and actually a former DePaul summer student of Bob Marrs. We just published a really cool paper, which we will be very happy to share, looking at mitochondrial dysfunction, right? Because you need mitochondria for everything. And we see in our AD neurons, and so I'll just kind of point to the relevant ones, just sitting there. We haven't done anything to these neurons. We are just measuring baseline calcium within the mitochondria, right? So mitochondria will take up calcium, they buffer it. It's necessary for bioenergetics, but too much is bad. And just sitting there, we have too much or a lot more calcium in the AD neurons. It's partially reduced by Ryanodex, but not entirely, suggesting there's probably more than one source of this. And there's channels that bring it in, this VDAC and this MCU channel are calcium channels. And we see, and then there's an extrusion channel called the NCLX. And what's really cool is they've shown that the MCU, the one that's bringing calcium in, is upregulated. So there's more channels to bring in calcium. And then the NCLX channel, the sort of exit pathway, is reduced. So there's less opportunity or less fewer mechanisms to release the calcium that's being pushed in by too much. And there's also a lot of that MCU. So again, really, really interesting cell biology that they're doing. But what is this calcium doing? That's always my question. Like, what's it doing? Why? Why does it matter? And we see in our AD neurons, again, just sitting there, those membrane potentials depolarized, and that has a lot, it's going to play into a lot of the electron transport chain and the function of creating ATP. It's even worse when we trigger that ryanodine receptor. This is also linked to increased superoxide production, so free radicals. So you hear like nutrition experts saying you need antioxidants. It's clearing these superoxides, these free radicals that are basically running around and wreaking havoc on other molecules, because it's disrupting their structure and function. And again, we can reduce it by normalizing calcium. And then we see increased caspase 3. Caspase 3 is involved in the cell death pathway, apoptotic pathway. And we know in Alzheimer's and many of these conditions, cells are dying when they shouldn't be. So like all of these things are essentially a downstream consequence to some extent because of this excess calcium. Like,
Right,
okay,
cool.
There's a lot going on there.
Yep. So there's a question about just some clarification before we get to Elise about were those cells from humans, or are those from that we probably came
Everything
from
I've been showing you, these are all from human
humans?
patients.
All right. And
These
then
are all humans.
Malhia, Malhia Islam asks, I'm a PA student, very exciting research. I'm a former reproductive biology researcher. My work was on prenatal brain development, comparing short versus long-lived mice to find neurodegenerative disease markers.
Oh, you should talk.
prenatally. We also worked in sex-biased epigenetic clock analysis. My question, was there any difference between male and female mice models?
Interesting. So we have dabbled in that, but not powered all of our studies to look at it. But to date, we have not noticed any differences in the male or female mice in these phenotypes. However, we do know that in the AD mouse, at least in our triple transgenic that we use, and also another one, a PS1 APP, the females had more aggressive pathology, like amyloid and tau.
Yeah.
Their calcium dysregulation was the same.
And as we get here closer to the top of the hour, I wanted to make sure we had a little time for Elise to talk about
Yep.
her work a little bit.
Absolutely, absolutely. And she works on Down syndrome stuff too. So how about I just skip? Basically, what I'm going to say is for the Down syndrome, we see all the same things.
Okay.
And this is human neurons from individuals with Down syndrome. But let's definitely have Elise tell us more.
Thanks so much for handing it off, and thanks, Dr. Sousa and DiMario, for the kind invitation. So my main focus in the lab is looking at how traumatic brain injury is a major risk factor for developing Alzheimer's disease, as Beth previously spoke about. Don't have to tell you too much about TBI. I'm sure we all understand. It's caused by a blow to the head, either direct or indirect. But like Beth said about AD, there's a lot we don't know about the consequences of traumatic brain injury and how it actually becomes pro-neurodegenerative. If you want to, yeah, there you go. Perfect. Okay, so a little bit what Daniel and I have been up to in the lab is much like the previous studies that we showed in the human cell lines, we're also looking at calcium dysregulation in our mouse models. We have a mouse model of repeat traumatic brain injury, and of course, that mouse model of Alzheimer's disease. And what's really cool is that you'll notice in A and B over here, we're looking at that source of calcium dysregulation. And much like in the previous studies, we're seeing an increase in calcium release from the ryanodine receptor on the endoplasmic reticulum in our repeat traumatic brain injury mice that mimics our Alzheimer's disease mice without a traumatic brain injury. So something is going on there that's driving that shared phenotype between Alzheimer's disease and repeat TBI from a calcium dysregulation perspective. And as Beth also alluded to, this has a lot of downstream consequences, one of those being in excitatory postsynaptic currents that are released by these neurons. And so this is really interesting because, again, in our traumatic brain injury mice and in our Alzheimer's disease mice, we're seeing a shared increase in the frequency of these spontaneous excitatory postsynaptic events. And this is a big impact on cell excitability and future potential of looking into, for example, excitatory and inhibitory imbalance in these animals as well that can also have behavioral consequences.
So help me with that. So if the cell is overexcited, what does that tend to do?
Sure. So while we know, for example, that when we're talking about calcium availability and this calcium dysregulation, if there's a greater calcium concentration on the presynaptic side, we're going to see an increase in synaptic vesicle release as well that follows that. And this is without any initiation of action potential, for example. So this is just a baseline overexcitable phenotype that could also have further consequences when we talk about, for example, a network level like the hippocampus, an area of learning and memory.
Okay.
And then, okay. So furthermore, when we're looking at the proteinopathy and histopathological staining of these animals, we're also seeing similarities in our repeat traumatic brain injury mice, once again, with our Alzheimer's disease mice when it comes to states of microglial activation and neuroinflammation, a really big topic in neurodegeneration right now, as well as that accumulation of phospho-tau and beta amyloid that Beth was also alluding to, which we all know are two major hallmarks of Alzheimer's disease as well.
So basically, in the control animals with a brain injury, it mimics phenotypically what an Alzheimer's control mouse looks like.
Exactly, which we wouldn't necessarily expect, considering that mice on their own, like as Beth was saying, don't generate Alzheimer's disease themselves.
So Hector Rosgato has a question. Changing intravesicular pH is likely to change free calcium concentration available for these release, because of course all of those little moieties that hang on to calcium are pKa sensitive. So if they've got enough hydrogen around, they won't bind the calcium. So he asks, comments? Is it really free calcium when it's in there?
So in the ER or the lysosome?
Yeah, sure.
Yes. So I know in the ER, much of it is bound to like calreticulin. There's calcium buffers. Probably within the lysosome, much of it is likely bound as well, though a lot less is known. Like they don't even know what the calcium influx channels are. They just know what the efflux channels are. So within lysosomes.
Because I mean, I guess the point would be right in serum, right? Almost all the calcium is bound up
Yeah.
on proteins of one sort or
Mhm.
another and not necessarily available.
Right, correct. So yeah, once it's in these organelles, it's usually buffered.
Okay.
But when it's released into the cytosol, it is then, quickly acting on whatever its target is, and then usually sequestered, buffered, or pumped out.
Okay.
So, do you want to go over the summary, Elise, or?
Yeah, sure. So just kind of tying it all together here, when we're thinking about what is the common feature that kind of binds these different pro-neurodegenerative phenotypes into each other and promotes that risk of AD, Alzheimer's disease, dementia, and neurodegeneration. And so, as we stated from our various models, we're seeing this increase in calcium release and dysregulation from the ranitidine receptor on the endoplasmic reticulum. So it's something of an intracellular dysregulation of calcium itself that's occurring in also Down syndrome, repeat traumatic brain injury, single traumatic brain injury as well by some older papers too, and that are shared between Alzheimer's disease. And as we've stated with the mitochondrial imbalances and the lysosome imbalances that follow, I like this graph a lot because it's a bit of a three-sided scale. You disrupt something somewhere, and somewhere else you're also going to see consequences. As we were stating, calcium kept at a really low concentration in the cytosol normally. So whenever it's released from an organelle, it really needs to be sequestered carefully before all kinds of things go awry. And what we believe is happening in these three states is that it's potentiating this neurodegenerative phenotype. So that's what we're going to continue exploring further. And I'm also really happy to state that we're also looking into some cool RNA sequencing data too regarding traumatic brain injury, especially what's going on in the mitochondria there as well.
Yeah. So, one of the things, free calcium is a problem, right? Essentially, rigor mortis is too much free calcium for the brief period of time, right? I mean, like that is, um, uh, so I, one of the tricky things about Alzheimer's and all of the various related neurodegenerative diseases is that for clinicians and family members, it's really hard to figure out which one of these things you have, right? So as you think about that and you think about your work, how should we think about the related conditions that lead to these, whether or not it's things like CTE, which is not really the same as the mild cognitive impair that somebody might have, for which we may not know the cause of, versus an Alzheimer's disease versus one of the genetic rapidly onsetting Alzheimer's diseases. Like how should we be thinking about this based on your work?
Yeah, so that's, I mean, a really important question. The bio sensors and blood-based biomarkers is blowing up in the field right now.
Yeah.
And they're largely measuring different types of amyloid and tau, which
And then trying to treat those, right? And
yes.
right, whether or not that's working or not is not necessarily it is a whole question, right?
Right. So I was more than happy to at least share my thoughts on some of the new targets.
Oh yeah, look at that. Yeah.
So yeah, so it's, I mean, I think, a lot of these present very non-specifically, like often some of the initial symptoms or changes in Alzheimer's present as depression or more of like an affective disorder or rage, something along those lines. And then it's not until later. So it doesn't all look the same. I'm a believer in, I think maybe we need to take two steps backwards and have a better understanding of what's actually the mechanism of the cognitive loss. That really doesn't seem to be as much of a priority as it is trying to understand amyloid and even tau, which have varying, correlations with memory loss in the disease. And then, would you treat them any differently? So frontotemporal dementia versus Alzheimer's disease. One's more of a tauopathy. Alzheimer's is more a little more global. Do you treat them differently? Like what's the outcome? And if we're trying to restore cognition, does it matter so much to get the exact diagnosis right in the beginning? And that's meant as a genuine question, not
Right.
or are we going to try and preserve the symptoms and go after that? And that hasn't been answered.
Yeah, and that's going to take us to the top of the hour. So
Yeah.
I want to thank Joe DiMario, Elise Weber, and Beth Stutzmann for being with us. I want to thank Jonna Purcell for helping us set it up and also for all of her incredible work to get the board meeting going this week. Jonna's had a huge week. I want to thank all of you for being with us today. Thank you for all you do to support our faculty, our staff, and our students. It is the work of people like you that make the university such a special place to be. It is Friday. I hope you get to have a weekend. Take care, everybody. Bye-bye.